There's one DNA fact that quietly breaks the simple story of European origins. Not because the story is false, but because it was never whole to begin with. What looks like a single beginning turns out to be two different genetic lines moving in different directions at the same time. And once you see that split, the idea of Europeans as one genetic people starts to fall apart. For a long time, Europe was described as a finished product, a place where populations arrived, settled, mixed a little, and eventually became what we now call Europeans. It sounds clean. It sounds resolved. But DNA does not tell stories that way. When geneticists stopped asking where people lived and started asking which line survived, the picture changed. Not dramatically at first. Quietly, piece by piece. This video is about rebuilding that picture from the ground up, not using legends or national stories, but using three kinds of genetic evidence that don't care about borders or identities. We'll look at the paternal line, the maternal line, and ancient DNA pulled directly from archaeological remains. When you place those pieces together, they don't merge into a single origin. They form a mosaic that never fully unified. By the end, you'll see why the word European describes geography far better than genetics, and why the people living in Europe today carry the legacy of multiple populations that never collapsed into one. Here's the strange part, and this is the thread we'll keep pulling. One genetic line points clearly toward the Eastern Mediterranean and the Near East. The other often doesn't. Sometimes it points deeper into Ice Age Europe. Sometimes it points somewhere else entirely. Both lines are real. Both are well documented. But they don't tell the same story. And that contradiction changes how we should even ask the question of who Europeans are. To understand why, we need to change how we think about origins. Not as a single moment, but as overlapping histories that left unequal traces. Genetics doesn't preserve everyone equally. Some lines spread aggressively. Others persist quietly. Some are amplified by migration. Others by isolation. What survives is not a balanced record, it's a biased one. And Europe is a perfect example of that bias. Think of this investigation like forensic reconstruction. The scientists aren't characters in the story. The methods are. The first tool is the Y chromosome. It passes almost unchanged from father to son, making it a powerful tracer of male ancestry. The second is mitochondrial DNA, inherited through mothers, which follows a completely different path through time. The third tool is ancient DNA, actual genetic material recovered from people who lived thousands of years ago, before modern populations existed. Each tool answers a different question. The Y chromosome asks, which male lines expanded and replaced others? Mitochondrial DNA asks, which female lines persisted through those changes? Ancient DNA asks, who was actually there before the later layers arrived? None of these tools is enough on its own. Together, they reveal something uncomfortable. Before we go further, there are rules to this reconstruction. No myths, no national pride, no assumptions about purity or continuity. Only population comparisons, migration patterns, and what the data allows us to say. Genetics doesn't care about modern identities, and it doesn't confirm comforting narratives. It only shows what survived. And here is where the first crack appears. If you only follow the paternal line, the story seems almost neat. Many dominant Y chromosome lineages in Europe trace back to populations that expanded from the Near East and surrounding regions during relatively recent prehistory. From that angle, it looks like a strong genetic anchor, one origin spreading widely. But that is only one line, and it is the loudest line, not necessarily the most representative. Because when geneticists looked at the maternal side, they expected the same pattern. They didn't get it. Instead, they found something messier. In many European populations, maternal lineages showed deep continuity with much older groups that lived in Europe long before those later migrations arrived. Lines that didn't expand outward. Lines that stayed local. Lines that don't point back to the same place. This isn't a contradiction. It's a record of unequal movement. Historically, large migrations were often male-heavy. Groups of men moved, settled, and integrated into existing communities. Their Y chromosomes spread rapidly. The maternal lines, already present, didn't disappear at the same rate. They persisted underneath the surface, even as the visible story changed. Ancient DNA confirmed this pattern in a way nothing else could. When researchers extracted DNA from skeletons spanning thousands of years, they saw clear genetic shifts, but not total replacements. Earlier populations didn't vanish. They were absorbed unevenly. Some of their genes diluted. Some remained stable. Some became rare but never disappeared. This is why the idea of a single European genetic people doesn't hold up. Europe was not formed by one population becoming many. It was formed by many populations layering over each other without ever fully merging into one. And that brings us to the question we've been circling without answering yet. If Europe never had a single genetic core, if its paternal and maternal histories diverge, then what does European ancestry even mean? Is it a place? A time period? A dominant layer that overshadowed others? In the next part, we'll start with the line that seems simplest, the paternal side. It gives a clean answer at first glance. A direction, a source, a sense of order. But that simplicity doesn't last. And once it breaks, the rest of the story becomes impossible to ignore. We're not looking for legends. We're looking for traces left in DNA. And the most direct place to start is the paternal line, the Y chromosome. It's the simplest genetic path we have, passed from father to son with relatively little change. If there were ever a single, clean origin story for Europeans, this is where it should appear. When geneticists first began comparing Y chromosome markers across Europe, something immediately stood out. Certain male lineages appeared again and again, across vast distances. From the Atlantic coast to Eastern Europe, large portions of the male population shared closely related Y markers. That kind of spread doesn't happen by accident. It points to expansion, rapid, directional, and recent in evolutionary terms. The clusters themselves tell the first part of the story. When you group European Y chromosome data, the strongest signals trace back toward the Eastern Mediterranean, Anatolia, and the broader Near East. Not everywhere, not exclusively, but clearly enough to form a genetic anchor. From this perspective, Europe looks less like an isolated continent and more like a western extension of earlier populations that expanded outward. To visualize this, imagine a map rather than a timeline. You start with a dense concentration of related Y lineages in the Near East. Then you see branches stretching westward into the Balkans, across Central Europe, and eventually to the Atlantic. Each branch represents generations of fathers passing on the same core markers, amplified as populations grew and spread. What matters here isn't the exact dates or mutation counts. It's the direction. The paternal signal doesn't radiate evenly from within Europe. It arrives from outside and becomes dominant over time. This pattern aligns with several well-documented prehistoric movements. Farming expansions, technological advantages, social structures that favored rapid growth. Groups carrying certain Y lineages expanded faster than others. They didn't just migrate, they replaced earlier male lines at scale. In many regions, older Y chromosomes virtually disappear from the record. From the perspective of the Y chromosome alone, the story feels almost complete. A strong incoming signal. Wide replacement. A continent reshaped by a relatively narrow set of paternal ancestors. This is why older genetic narratives leaned so heavily on this line. It offered clarity, but clarity can be misleading, because the Y chromosome doesn't tell you who was already there. It tells you whose sons survived in greater numbers. Replacement doesn't require extermination. It only requires imbalance, differences in fertility, status, or mobility that play out over centuries. The genetic result looks decisive even when the human reality was gradual and complex. Ancient DNA makes this clearer. When researchers analyzed skeletal remains from Europe before and after these expansions, they saw sharp shifts in Y chromosome composition over surprisingly short periods. Entire male lineages vanish from later samples. New ones take over. On paper, it looks like a reset, but the rest of the genome doesn't reset the same way. This is where the anchor metaphor matters. The paternal line gives weight and direction, but it doesn't stabilize the whole structure. It holds part of the story in place, while other parts drift. If you only followed the Y chromosome, you could reasonably conclude that Europeans descend largely from a small number of Near Eastern, adjacent male populations that expanded into Europe and reshaped it. And many early interpretations stopped there. The narrative felt resolved. But genetics doesn't reward stopping early, because while those paternal lines spread, they didn't spread into empty land. They moved into regions already populated by groups with deep local histories. Those encounters didn't erase everything. They reshaped who passed on what. The Y chromosome is especially sensitive to this kind of change. A relatively small number of successful males can transform the genetic landscape in a few centuries. Their markers multiply quickly. Older lines fall below detection thresholds. Over time, dominance looks like origin. This creates a dangerous illusion. The illusion that the most visible genetic line is the most important one. It's also why the paternal story feels so comfortable. It mirrors how history is often told, through expansion, conquest, and replacement. It gives us a clean arrow pointing backward to a source. It answers the question, where did they come from, in a way that feels decisive. But even here, there are limits. Not all European Y chromosomes trace to the same source. Not all regions show the same degree of replacement. Some pockets retain older paternal diversity. Others don't. The anchor holds, but unevenly. And more importantly, this anchor doesn't tell us who Europeans were. It tells us which male lines became dominant. That distinction matters, because when you step back and compare the paternal signal to the rest of the genetic evidence, you start to notice gaps. Places where the Y chromosome tells a story that the rest of the genome doesn't fully support. Places where continuity exists alongside replacement. If we stopped here, the story would be neat. Europe as a genetic outcome of successful male expansions anchored in the Near East. A single dominant trajectory branching westward. Many people still hold this version implicitly, even if they've never heard it stated this way. But this neatness is exactly what should make you suspicious. Because the moment you switch tools, when you stop following fathers and start following mothers, the anchor loosens. The direction changes. The timeline stretches backward. And the idea of a single European genetic people becomes much harder to defend. That's where the investigation goes next. Now we change tools. We stop following fathers and start following mothers. And almost immediately, the geography shifts. Mitochondrial DNA doesn't point as cleanly toward the Eastern Mediterranean. In many European populations, it sinks deeper into the continent itself, sometimes far deeper in time. The maternal line doesn't echo the paternal story. It complicates it. This is where two truths collide. On the paternal side, many dominant lineages arrive from outside Europe during later prehistoric expansions. On the maternal side, a large portion of lineages show continuity with populations that were already present long before those expansions began. These two facts sit side by side. They don't cancel each other out. They describe different movements happening unevenly. At first glance, this looks like a contradiction. If people arrived from elsewhere, why don't maternal lines reflect the same origin? The answer isn't mystery. It's mechanism. Large-scale migrations in prehistory were rarely balanced. They tended to be male-biased. Groups of men moved, settled, and integrated into local communities. Their Y chromosomes spread rapidly because male lineage replacement only requires demographic advantage, not total population turnover. The women they partnered with, however, often came from the existing population. Their mitochondrial DNA stayed local. Over generations, this produces a very specific genetic signature. Paternal lines shift quickly. Maternal lines change slowly. The result is a population that looks new from one angle and ancient from another. Ancient DNA makes this visible in a way modern samples alone cannot. When geneticists examine skeletons from different periods in the same region, they often see continuity in mitochondrial lineages across major cultural transitions. Farming appears. Technologies change. Languages shift. But many maternal lines remain surprisingly stable. This doesn't mean nothing changed. It means change was layered rather than absolute. Earlier populations weren't erased. They were absorbed asymmetrically. This is also where the story stops being abstract, because these patterns aren't evenly distributed across Europe. In some regions, maternal continuity is especially strong. In others, it's weaker. Geography matters. Isolation matters. Local history matters. Think of DNA like language. You can replace an official language without erasing every accent. Vocabulary shifts faster than pronunciation. Power changes faster than habit. Genetics works the same way. Some lines adapt quickly to new structures. Others persist quietly underneath. This analogy helps explain something else geneticists noticed. In populations that experience repeated waves of incoming males, maternal diversity often remains higher than paternal diversity. Fewer Y lines dominate. More mitochondrial lines coexist. That imbalance isn't random. It's the residue of repeated integration, not repeated replacement. There's a biological consequence to this pattern, and it's one researchers approach carefully. In isolated communities where a small number of founders contributed disproportionately to future generations, certain genetic traits become more common, not because they're advantageous, but because they're inherited from a limited pool. This is known as the founder effect. It's not a story about deficiency. It's a lesson about how history shapes biology in quiet ways that matter for medicine and population studies. But the larger implication is conceptual. If European populations carry paternal signals, pointing toward one set of origins, and maternal signals, pointing toward multiple others, then the idea of a single genetic identity collapses. What exists instead is a record of interaction, movement layered onto continuity. And this mixing wasn't uniform. Some regions saw more incoming influence. Others retained more of their earlier structure. The result is not a blended average, but a patchwork. This is where modern identity often runs ahead of the evidence. We talk about Europe as if it formed once and diversified later. The genetic record suggests the opposite. Europe was always diverse. What changed was which layers became dominant at different times. The maternal line preserves that memory better than any written source. It carries traces of populations that left no monuments, no texts, no recorded names. Their presence survives not in stories, but in inheritance. And this leads to the next question we haven't answered yet. If this pattern holds across much of Europe, what happens in communities where geography, isolation, or tradition change the rules entirely? Places where mixing followed different paths. Places where neither paternal nor maternal lines follow the continental pattern. That's where the story goes next. Once you step outside the main European corridor, the pattern becomes clearer, not weaker. The same genetic tools applied elsewhere show that Europe was never unusual. It was typical. Human populations almost never form as single genetic units. They form through layering, contact, and uneven inheritance. In regions like the Horn of Africa, South Asia, or East Asia, geneticists see familiar structures expressed differently. Paternal and maternal lines don't always point to the same places or the same timelines. Some populations show deep local continuity with limited external input. Others show repeated overlays from neighboring regions. In each case, identity emerges from history, not from a single biological origin. What matters is not comparison, but principle. Genetics reflects movement, not meaning. A population can maintain a strong cultural identity while its genetic components remain diverse. Language, ritual, and shared memory often stabilize communities long after genetic inputs have shifted. Europe followed this same rule. Its culture solidified even as its biological layers remained multiple. This brings us back to the central idea of the video. Europe was never a single genetic people. It was a meeting ground, a place where different populations encountered each other across millennia, sometimes merging, sometimes coexisting, sometimes replacing specific lines without erasing entire groups. What survived was not purity, but persistence. The genetic mosaic is not a metaphor, it's a measurable reality. Ancient DNA shows it. Modern population comparisons confirm it. The paternal line provides one axis of movement. The maternal line provides another. Neither is complete on its own. Together, they describe a history that cannot be reduced to one origin or one people. This is why arguments about genetic essence fail so quickly under scrutiny. The data doesn't support them. Identity is not encoded in a single chromosome or a single migration. It's distributed across thousands of years of interaction. And this is where we close the loop from the beginning. We started with the idea that the simple story of European origins was incomplete. Now we can say why. The mistake wasn't in saying people migrated. The mistake was in assuming those migrations produced a unified genetic outcome. They didn't. They produced a layered one. European ancestry isn't a point on a map. It's a stack of histories. Some recent, some ancient, some dominant, some nearly invisible unless you know where to look. Genetics doesn't replace history, and history doesn't override genetics. Each limits the other. DNA can tell us where lines moved and which survived. It can't tell us who people believed they were. Written sources can tell us how communities understood themselves. They can't tell us whose genes endured. Identity exists in the space between those two. If there's one thing to take away, it's this. The past wasn't simple, and it never needed to be. The idea of a single European genetic people is a modern shortcut, not an ancient reality. If you want to see another DNA story that breaks a clean origin myth, follow the channel.